Welcome, everyone. We're so happy that you've all joined our session today. My name is Joan Damerow, and I'm one of the co-chairs of the Physical Samples Curation Cluster. I'm joined also today by Andrea Thomer, who is here in the room, and Val Stanley, who has joined remotely, and she'll be monitoring online participation, so questions in the chat and whatnot. Chad Linktoe is here as well, and he is our ESIP Community Fellow for this cluster, and he's also here to help in this session. So what we want to do today is address this general question, the title of our session: how can we connect and track use of physical samples and associated data? So what is the basic guidance that we can provide to researchers, and what efforts and tools are being developed already to track sample use, and what are the next steps? There's still a lot of work that needs to be done. So this encompasses a general area that we've been working on in our cluster for some time now, and a continuation of some of our previous discussions that we've had in this cluster. Just a note here, we do have session notes available. There is a QR code that you can use to access the notes, and the link is shown here if you want to access that. I'll give you a moment. And in the session notes, if you can enter your name and affiliation and contact information, that would also be great. And you can also feel free to contribute to the notes. So if you think something should be added there, or if you have a particular feedback or question that you think you'd like to put in the notes, please feel free to do that. It's for everyone to contribute as needed. Just to provide you with a little bit more background for those who have not participated in our cluster previously, the Physical Samples Curation Cluster was established just a few years ago to support people in ESIP who are working with physical samples. So this can really be anything from like geological cores to voucher specimens and much more, things that you're out there collecting in the world. And so when we started this cluster, and we were gathering feedback on what people wanted to address and what tasks we could focus on, Shelley Stahl from AGU, who just walked in, she mentioned that there's a need for general guidance for AGU authors who are dealing with physical samples and associated data. And we kind of agreed as a cluster that this was something that was missing. We need some general guidance for scientists and researchers dealing with physical samples that's applicable across fields to make samples more fair. So the cluster has been working on the general guidance for authors, and we'll be elaborating on this guidance in an associated paper that we'll discuss in a moment, and that we'll invite you all to contribute to. So we are getting close to a final draft version of this, but one of our goals for this session is to get some additional feedback on what we've done so far and encourage more of you to get involved and help us refine the guidance. As a cluster, we're also now planning to do a variety of both working sessions. We'll be working on the paper, for example, and then also having some of our cluster meetings focus more on presentations and discussions. So we generally meet twice a month on alternating times currently. And I wanted to point out that we have a couple of interesting talks lined up for August 22nd. Rory Edmonds will be presenting on DataCite samples, related identifiers, and metrics, and we'll also have a discussion with him. And on September 12th, Neil Byers and Chuck Parker from the U.S. Department of Energy's Joint Genome Institute, they've been doing a lot of work on genomic sample citation and how researchers are citing genomic samples in the literature and how they are using this data in the literature. And they've also developed some associated software to extract those citations. And so we'll hear from them and have a discussion then. We also have plans to start co-hosting some of our meetings with the System for Earth Sample Registration to have discussions on digital and physical curatorial challenges for samples. So these are some of our plans, and if you're interested in getting involved, we would love to have you, and you can join our mailing list to get more detailed information on our upcoming meetings, and we'd be really excited to have as many of you join as who are interested. There are a couple of related announcements. I'm co-hosting an AGU session with Aaron Robinson and Leslie Wyborn, and also Rory Edmonds on enabling open and interoperable access to physical samples and their associated digital artifacts. So this would be a really great group. Any of you who are interested in submitting an abstract to this session, we really encourage you to do that. The deadline for that is August 2nd. Something else that came to our attention recently, there is a public review going on now of proposed changes to Darwin Core by the Material Sample Task Group. So some of you might be interested in that, and we have a link in the slides for that. Throughout the session today, our goals are to, well, the numbers got messed up there, but first present sample sharing recommendations and get your feedback. We'll then, through our presentations, learn about some efforts to use sample identifiers and metadata and workflows and to track sample use. We'll then discuss some of our priorities and next steps to better enable sample use tracking in the future. And then what we want to do after this is to use some of what we learn in the session and then work beyond this session in an associated paper, which would include these guidelines that we'll discuss today, along with additional information on use cases and current efforts and needs for sample use tracking. So we have a couple of ways that you can contribute. We have a link here for our current draft of the sample sharing guidelines that you can take a look at and review more closely later. Andrea has also developed a form that includes all of our it includes a checklist of the basic recommendations that we have in our guidelines, and that's what we'll review today. We're going to review this checklist of some of our recommendations, and so you can follow along in the form. We encourage you to do that, but you may not have time to fully complete the form. We'll give a little bit of time to think about it and respond to what you can in the form, and then we'll move on to the next thing. And we hope that if you don't finish the form in the session, that you could do it really quickly afterwards, after attending this session today. And we'd love to get all of your feedback, so we can have more representation in these guidelines and refine them. So I want to make sure that we're all on the same page for what we want to do with these sample sharing guidelines. We have discussed this fairly extensively in our monthly cluster calls, and this is what we have for our goals. We want to provide some basic information to the general scientific researcher on how to describe and reference samples in their journal publications, and keeping in mind that what we're using the currently available infrastructure. We may not have everything in all the ideal tools that we wish we would have. The second is to make samples and associated data more fair. We want to make it possible for physical sample repositories and collectors to get credit when samples are used. We want to make it possible to find and access physical samples that are used in a study that you may be reading. And overall, we want the guidance to provide information on what we need, the foundational elements that would enable more sophisticated, complex citations, and or to track use of samples across publications in the future, potentially. or even using automated tools today. So this includes recommendations for sample identifiers and cross-domain metadata that would be needed for a sample citation. All of the information that you might need to find, access, and give credit for samples. And so we've distilled our recommendations into a checklist for how to plan and publish research that includes physical samples. And I'm going to walk through that now. And if you are able, you could follow along in the form. If you're not interested in doing that, you can also just follow along with the slides and jot down any notes that you have or, questions. So step one, this would be on the second page of the form, I believe. So you can skip over the first page if you are following along and you haven't done that yet. Has everyone been able to access the form who wants to? I can go back to that link if needed. Yeah. Should I go back?
The link is also in the notes document for this session if you're in Pico chat.
And you can skip over a lot of the text and just follow along and, revisit things if needed. Okay. So step one, we're recommending that you use identifiers for samples. So if your samples already have persistent identifiers, use them in associated papers and data sets. We do know that a lot of samples that actually do have PIDs are not actually being cited in data sets and papers. And so we want to make this clear. If your samples already have PIDs, find out about that and use them in associated papers and data sets. So to do this, work with your institution's sample repository, laboratory, or data repository manager to obtain these existing PIDs or PIDs generated upon submission to associated databases or systems. And if you're taking subsamples of a sample with a PID, obtain a new related PID from your institution. In many cases, in most cases, samples may not already have PIDs. If this is the case, obtain PIDs from an external organization like the System for Earth Sample Registration that provides IGSNs, or EasyID that provides ARCs. And there is John Coonsey, who is here in our session today, is holding a session on ARCs tomorrow, if anyone's interested in that. If you can't obtain a PID, use a reasonably unique identifier, like a universally unique ID, a Darwin core triplet, or maybe even just a project-specific unique ID. Some people might disagree there, but that's what many people are doing. And we wanted to point out in the guidance as well, we had some discussions on when do you need PIDs for samples. And we think they're most important for samples that are in long-term collections that you don't know how they're going to be used. They're ideally going to be used over time. And or they're used in multiple analyses, data systems, or papers, for example, in highly interdisciplinary work. So I'm going to stop. I'm going to take just some brief moments so that you can look at the form and respond. Or think about any questions that you have regarding this, or any brief comments, you could bring them up. Or save them. We're going to have a time afterwards for questions. We have a couple questions.
Have you considered including possible RRIDs as well as a unique identifier?
Yes, we could definitely do that.
Okay. Do you want me to put that in the form?
Yeah,
okay.
That would be great. Yeah, any any additional things that we need to add, that would be great, a great thing to add to the form, or maybe even our notes if you're not accessing the form. Okay. And
Joan, we have a question in the chat as well from Steve Richards. Who he says, sounds like PID in this context, sorry, gotcha, means a web resolvable permanent identifier.
yes, and in the guidelines, we do have probably too lengthy an explanation of PIDs, but we're just going through the checklist right now. So one of the things that we will actually need to do is work on simplifying. But yes, did you have any issue or
I just
comment?
wanted, a lot of times when people use PIDs, they're not necessarily talking about resolvable identifiers.
Yes,
Just means-
that's what we're talking about. Okay, I'm going to move on, and then so if you do have additional questions, we can save that for the end. We're also working on other ways of presenting the information. So we're kind of experimenting with flow diagrams. I know you can't read this, so I'm not going to go through this. It's basically a flow diagram of what kind of ID you might need. The second step is to describe samples, and this slide is just giving some background information on cross-discipline metadata, which really comes from the work of the Internet of Samples, and then also just basic metadata, like from bibliographic metadata from DataCite. And so we have a list of the metadata that we believe, based on previous work, that is most useful for global sample search and for giving credit for samples. But if you have additional, if you think additional things are needed, then that would be a good thing to put into the form or to contribute to our guidelines. So I'm going to move on to the next one here. So what to do here for describing samples is to create a standard metadata file or table in your paper that describes the basic characteristics of the samples that are used in the paper and how they were collected and archived if they were archived. Ideally using a domain-specific metadata template. And we provide some examples here of domain-specific metadata template. These are all typically spreadsheet templates with just the basic sample variables, sample metadata variables. The similar information to what I showed on the last slide with additional requirements based on that particular discipline. And so if you have any recommendations on additional templates that we should add there, that would be a really great feedback to give in the form. So again, I'm just going to pause for a minute to give you time to address the form. Okay, I'm going to move on now. The next step is to archive the physical samples and metadata. So archive samples that are not destroyed. If you have access to a physical sample repository, archive your samples according to the requirements of that repository. If you do not have access and your samples were not destroyed in analyses, consider preserving them for some time according to any funding or institutional requirements. And associated with that, archive your sample metadata describing these physical samples. So the recommendations that we've come to here are, if you use less than 10 samples in your paper, you might want to just include a table with your sample metadata in the paper, either in the methods section or potentially in a sample availability statement. And we have some examples of doing that in the actual guidance. If you use more than 10 samples in a paper, archive a file containing the standard sample metadata as part of a data set and reference that data set in your paper. If you obtained sample PIDs the sample metadata may already be archived and associated with your individual sample PIDs. So in this case, you could opt to simply reference the PIDs in your paper. Archive the sample data. This is a continuation of what I was just discussing. So publish data associated with your samples in a domain or institution specific data archive. And we give some examples there. Additional examples that you think should really be included here, put those in the form. If there is no relevant archive, you can use generalist repositories such as Dryad, Zenodo, or Figshare. And then we have a few specific recommendations for what should be included in the data set with your sample data when you publish that in a data repository. So as I mentioned, when talking about metadata, we want you to include a standard sample metadata file that describes each sample using a domain-specific template, if possible. And include a list of the sample PIDs in the appropriate standard format within your data set metadata. All that information that describes your sample data set, the full data set. The sample metadata field, or the specific metadata field, data set metadata field that you use to provide these sample IDs may vary depending on the data archive. But it could be a field specific to samples or a free text field such as the methods. And the reason we have this here is to help ensure that sample PIDs could be searchable when you're searching data sets, because most repositories don't enable search within data files. And so we want those PIDs to be in the data set metadata. Then all sample related files in your data set should consistently use the appropriate sample identifier, such as within a column or field for the sample ID or IGSN. And then finally, reference the samples used in your paper. So include sample identifiers in the text and or within one or more tables in your paper, such as in the methods section or supplementary material, which could still be problematic. So if you have any recommendations there, put it in the form or let us know. And so we want to provide the sample PIDs in their standard format with hyperlink, and we provide examples in the guidance. And here there's an example of an IGSN in its standard format with a hyperlink here. If using other identifiers, list them consistently in your paper exactly as they appear in any associated data sets or files. Do not use approximate variations of the same identifier. And then cite your associated sample data set or maybe individual samples. So the sample data set can be cited in the data availability statement or the references section. And then if you have just a very few number of samples, which could be the case for some papers, they could be cited in a data availability statement individually, particularly for a small number of samples like I mentioned. And again, the purpose of this, what we want to do is to help ensure that potential data users can access the physical samples used and help provide credit to the original sample and data collectors, as well as curators of samples in the physical repositories. So that's what we have for our guidance, the checklist, and we really need some more feedback. This is based on work in our cluster meetings, and it would be great to get some additional contributions and feedback there. So again, we have the link to the for you to contribute to the guidelines, and anyone who contributes here would be included on a paper that we hope to submit in the next few months. And then you can also, we're hoping that you'll finish providing feedback in the forum if you've been following along with us today. And so we have the link here again for that. Okay, so now we're going to move on to the invited speakers for this session. Actually, do we have time for any questions before we move on? Or okay. Okay, so we're right at time, unfortunately, so we don't have time for discussion now, but we will at the end. So if you do have some things to bring up about this guidance, we can do that at the end after our presentations. So the invited speakers for this session are Andrea Thomer, who will be presenting on obstacles to showing the impact of natural history collections, Dave Villaglace, who will discuss citing samples and the Internet of Samples, Daniel Noscart, who will discuss tracking the use of biodiversity data in the Global Biodiversity Information Facility, and Alicia Wood-Charlson, who will be presenting on sample data and identifiers in the U.S. Department of Energy Systems Biology Knowledge Base. And so the next speaker is Andrea, who's here.
Great. Hi, everybody. I wanted to say one last thing about that the survey. There's a little bit at the end where you can put in your email address if you're willing to be bothered directly for future involvement. So if you're willing to help us develop examples, or you want to contribute to the paper or anything else, please share your email. And if you have feedback about the survey itself, please give us that too. Like if it's too long or confusing, because we plan on sending this out more broadly to ESIP and to other folks that aren't here right now. So please let us know if something looked weird. So I'm going to try to keep my talk to an actual 5 minutes, and Chad is going to help me stay honest, because I gave my other 5 minutes to Dave. So I wanted to go first because I'm in person and that's just easier, but because this is going to kind of frame why this is a problem. So if you haven't been to one of these sessions before and you're trying to understand why do we need sample citation guidance, I think this project helps illustrate this. So this is something I've been working on for a couple of years with a bunch of collaborators, notably Sarah Lafia, who was a community fellow last year. And this started out as trying to mine specimen citations so we could show the impact of natural history collections. And it turned into a paper on why it's hard to show the impact of natural history collections. So the reason for this is that natural history collection specimens, and importantly, their stewards, the collections managers and curatorial assistants that are actually doing all this really hard work of keeping these collections accessible, are rarely recognized as being as important as they are. They don't get a lot of funding. It's just kind of like this precarious position that they're always in. So working with Cody Thompson at the Museum of Zoology Mammals Division, we started a project trying to figure out how we could show the impact of these collections similar to the way that we show the impact of our own research. We eventually found some collaborators at the Smithsonian Museum of Natural History that we started working with too. The reason we worked with these two collections is that both of these collections have bibliographies that their curators have been, or collections managers have been trying to hand curate over the years of papers using specimens in those collections. So Cody's been doing this by hand. He maintains a Google Scholar page for the mammal division. And it includes, and this is important, not just papers that use the specimens, but also papers by researchers that work in the museum. So it's a little bit of a mixed bibliography. The Smithsonian bibliography, I believe, is mostly created through Google searches, so it's a little less refined. But again, it is at least a good starting point of papers that we're pretty sure have to do with these collections. So in order to mine references, we were looking for specimen identifiers. And most of them, these are older papers. They're not going to be using PIDs. They're going to be using something more like a Darwin core triplet. So an acronym for the collection plus a random number indicating what specimen it is. And we tried 3 different methods to mine this information out of the papers. So first, we just tried regular expressions, so pattern matching. Second, Sarah and Kat created some custom named entity recognition models trained on some data that we tagged. Oh God, I already have 2 minutes. Okay. So it was trained on data that we tagged. And finally, we tried using ChatGPT because everybody keeps talking about how that's going to revolutionize everything. And we wanted to see if that was actually true. The challenge here is that While there are some explicit references where there are actually the specimen identifiers in here, by and large, as we found, most of the references to specimens are vague. We looked at a bunch of skulls from this collection, or we thank this collection in the acknowledgements, but they don't say what they actually used from the collection. And that is the biggest problem that there is in doing this work. And that's why we're making this guidance, to try to get researchers to put this information more explicitly into the papers. Very short little bit of our results. The main thing we found, we were able to mine some citations. And so this is showing what we found from the University of Michigan zoology bibliography. We found some, but it was just not as many as expected. So the main takeaway here, so this graph is showing our most cited specimen, which is a Myocetus astragalus, which is not, first of all, not even from the Museum of Zoology, it's from the Museum of Paleontology. But second of all, it's only showing up in 14 papers. Compare that to the number of citations that we have for just the papers in Google Scholar. So UMMZ has almost 90,000 citations overall, and their top paper has 13,000 citations. Granted, these are very old papers, and granted, this is a messy bibliography, but we think this is too far of a span. We're still missing a lot of data. I wanted to present this because it helps show why we need to do all of this work. We found that all of our text mining methods have different kinds of shortcomings, and I wanted to emphasize, including ChatGPT, you have to do a lot of careful prompt engineering in order to get that sort of system to work. Another problem we had is that most of the legacy literature just isn't accessible. The PDFs aren't findable, or you can't parse them, so that the text comes out all garbled. A lot of data is hidden in various things that we were not able to programmatically download. So stuck in supplementary files or appendices, or in files that were in other locations, or in tables that we couldn't parse, or in the captions of figures that we couldn't actually get the text out in a good way. So this is kind of our last kind of discussion points for thinking about how to go forward with this. How do we make sample citations more mineable based on this work, and how to show the impact of collections? We need to require sample citation in papers. We need to somehow keep those sample citations with the papers, either through links that you can follow, or by keeping them in the actual text of the paper somehow, whether in metadata or as part of like the references section. All of that metadata for sample citations needs to be exposed by the publishers and in a harvestable way. And on top of that, we need infrastructure for aggregating citations, moving towards open access publishing, as we've all been talking about here for decades. And I'm going to talk about this at the end. I think we need to allow collection stewards to contribute curated bibliographies to help build the critical mass of knowing what papers are connected to specimens. And I can talk about that later if anybody's interested. I went over time, I'm sorry. Okay. All right. I don't know how to switch to the next talk. Yeah. Stop sharing. Sweet. Great. Oh, it is.
Okay, can you see my screen?
No. Give us just a minute.
But it's on Zoom.
It's on Zoom. It's just not showing up on the screen here, which we're trying to fix.
Okay.
Okay. There we go. Okay. Yeah, now we're good.
Okay. Hi, everybody. Sorry I can't be there in person, but it sounds like a really interesting meeting so far. So I'm going to talk just briefly about an approach that we're taking in iSamples, the iSamples project, which is an NSF-sponsored project. We're in our, almost in our 4th year now. And there's multiple institutions involved. And really, the iSamples project is really about facilitating access to information about physical samples. And we're trying to be collection agnostic, or, the type of specimen we're not really concerned about. We're more concerned about any physical specimen. How do you access metadata and promote access to that metadata and discover it? So we cover everything from, biological specimens in Smithsonian through to cores and so forth at CSAR, and pretty much everything in between. Everything that we access in iSamples has a globally unique resolvable identifier. And that's super important in the iSamples project, because what we're trying to do is promote a general pattern of access via linked open data paradigms. And so basically that means that every identifier has every record has an identifier that you can resolve to the information about the physical sample. We built the infrastructure to be agnostic with respect to identifiers, so ARCs or IGSNs or DOIs, etc., may be used to refer to a specimen. And then the individual records may have components to them, which are also referenceable by different types of identifiers, such as ORCIDs for agents or people and so forth. And so this is conveniently lets you do all sorts of interesting things with individual specimens, like you can just see a map of, this is Morea, Tahiti, with individual specimens. The towers of points here are individual specimens that have been recorded from the same location. Each point has an identifier. You can click on that point and see the individual record. Information. If you take a look at the sample record itself, it's really composed of multiple components arranged in a graph type of structure, where the physical sample record itself is kind of the root, and then we have all these other pieces hanging off that root instance. So, for example, an agent may be a collector, and the sampling event is another part that's attached to the physical sample record. And the interesting thing about this approach is that you can have different views across all the sample records, so that you can restrict the information that's shown depending on the sort of use case. So, for example, in a sort of general view, you want to see all the information. And so in this case, the sample identifier refers to not just the sample record, but basically the entire thing. Right? So all the pieces that are directly attached to that physical sample record. Similarly, if you take just a spatial view of things, then you may want to restrict the predicates to, the sampling event and associated information there. And so we're able to exclude the other pieces of information for efficiency reasons, and also just, just depending on the purpose of the content. Similarly, if you have a provenance view, then you may want to see the sample record, but also all the related records through the provenance chain to the origin record. So this is really useful for tracking subsamples or analyses and so forth. In supporting this sort of functionality, there are some really basic fundamental operations that are required by the infrastructure. And the number one thing is that you can resolve identifiers reliably to a chunk of metadata that's machine parsable. That's super important. But also we need to be able to gather this information in a way that is consistent in the representation of types and so forth. But the benefits of this, so there's some downsides, meaning that you have to have extra infrastructure to support access to this information, but it's pretty basic stuff. And the benefits of this is that there's all sorts of opportunities for leveraging these resolve operations to provide things like gathering metrics for access to and reporting, and also opportunity for restricting portions of these composite records based on, if there's sensitive information that may only be accessible for particular, people, then we can restrict that. Well, there's an opportunity to be able to restrict that. Now, what if we consider a view from a citation perspective? What do you need to present or capture in a citation in a publication. Really, what it comes down to is that you just need the unique identifier for a sample record. And then if you can rely on the infrastructure to resolve all the pieces of that sample record, then anybody seeing publication and identifier in there can, of course, resolve that to construct whatever view of the record they choose. Then expressing, we've already gone over this, Joan captured some of this information earlier, but just to reiterate, presenting, capturing or representing samples within a publication, there's multiple ways to do it. The simplest, of course, is to just simply list the identifiers in the publication, but there's obvious practical limitations. One interesting aspect of this, though, is that there's a very clear association of the context of use of those sample identifiers. So you may have a section where you say, well, we didn't use these samples for some particular reason. And if you just have a list of identifiers for samples that includes things that you didn't use as well as that you did use, then you lose that context. The next obvious approach is to use what's being termed a Relic Area document, which is basically a set of identifiers with some context associated with them. This is nice because it's essentially unlimited in size. You can add as many records as you want. You can add as much context information as you want. And this Relic Area document is really, when it comes down to it, it's a graph of information that can be similarly resolved to build up the pieces of the sample records that you're looking for. And then, of course, you can sort of expand this a bit and have like multiple levels of indirection where you can have relic areas pointing to relic areas which point to samples. There's all sorts of interesting uses for this approach, but we don't need to get into the details now. So in iSamples, we do have prototype relic area support, which basically enables users to select a set of records either by a query or manually, and generate a document which contains the list of identifiers plus some basic information about it, including the query that was used to generate that list. This is by no means a novel approach. It's been used by GBIF, for example, and other systems, but it's very powerful in that you get both the static list of identifiers and samples that match your queries, but you can also revisit this list later and re-execute the query and see what new stuff has changed, will show up. So in summary, there's a couple of key points that I'd really like to emphasize here. One is that this relic area approach is very powerful and it's a very useful mechanism for providing or sharing, a common known set of identifiers to samples. And then with some basic tooling, you can take that set of identifiers and expand it to all the metadata about those samples. Format that is useful to consume. One of the downsides of relying on this kind of infrastructure is that service implementations need to be consistent. And so that'd be a great recommendation for any sort of infrastructure supporting this stuff. And then there's also, anytime you're dependent on a service, you're introducing some fragility in that services may go away. So it's something to be aware of. And I went a little bit over time, I think, but thank you for your attention.
All right, I think it's my turn.
Yes.
Okay. And can you see my screen? I can't really keep track of the chat, so I'm just hoping you can actually hear and see me.
Yes.
All right.
We can hear you.
Excellent. Thank you. Thank you. Great. Okay. So yes, I'm Daniel Noscart. I work for the GBIF Secretariat, the Global Biodiversity Information Facility, and I'm here to talk about how we track the use of biodiversity data in GBIF. So for those of you who don't know GBIF, we're an international network and data infrastructure aimed at providing anyone, anywhere, open access to all data about all types of life on Earth. And our vision is a world in which the best possible biodiversity data underpins research, policy, and decisions. And our mission is to mobilize the data, skills, and technologies needed to make comprehensive biodiversity information freely available for science and decisions addressing biodiversity loss and sustainable development. So in terms of what data is in GBIF, I just pulled some numbers yesterday. So GBIF is basically a big database of where species occur in time and space. We have about 2.3 billion records of such occurrences across around 89,000 data sets. These data sets are shared in GBIF by about 2,000 publishers, and we are funded by a large group of countries, and we also have participation from a lot of international and thematic organizations. GBIF data gets used a lot, and yesterday we were up to about 9,000 confirmed cases of use of GBIF data in peer-reviewed papers. So data gets used across a variety of different topics, including human health, climate change, invasive species, food and agriculture, and so on. And we're looking at about close to 5 new peer-reviewed papers using data from GBIF being published every single day. So how do we actually track the use of GBIF data? So first off, all data sets in GBIF are assigned a DOI. So everything I'm going to talk about now is at the data set level. And so we're not actually talking about the individual records, just to make that clear. But when users search for data on GBIF and they find what they're looking for and download that data, that download or the search result are also assigned a DOI. Any data that is obtained through another source, whether it's a synchronous API call or through a software library or a third-party tool, can also be assigned a DOI later on, assuming that they have saved the necessary metadata. But the bottom line is that all data that comes from GBIF can and should be cited using a DOI. These DOIs, when we're talking about downloads, resolved to a landing page that was also mentioned by Dave in his talk. So the download landing pages contain information about the filters that were used to generate the download. It also has links to all the parent data sets and their institutions contributing records to the particular download. And it gives anyone the ability to re-download the same data. And we then have a fairly extensive and still quite manual effort into literature tracking. And this is based on part metadata from Crossref data site and so on, but also to a very large extent information coming from Google Scholar and directly from journal publishers. But through that, we're able to record citations and we log them in our own database, and we also feed those citations back to DataCite. In our own system, we unpack citations of downloads so that we can contribute citations toward the individual data publishers whose data was used in a publication. So I'm just going to break for a second and give you a quick demo of what this actually looks like. See if this works. Click that link, stop sharing, and then I'm going to share a different window. Let's see, this one. All right. So you should be able to see my browser window now, I'm hoping. So What we're looking at here is a paper that was published just a few days ago. This paper, if we go down to the methods section, have used flowerfly locality data, and some of these records came from GBIF. There is a reference to GBIF here. If we click that, we can see that here is indeed a GBIF, occurrence download, and there's a DOI. If I click that DOI, I come to the download landing page, which will show me the details of the data that was included in the download, in this case, 65,000 records. You can see which filters were applied. So we have some geometrical filters. There is a scientific name of the taxa that were involved in this download. And there's also some filters on issues. Anyone coming to this page will be able to see that. They'll be able to re-download the data, and they can also rerun the query on current data. So we can see here that this download from June 2020 with 64,000 records, if rerun now, would have considerably more data. Most importantly, if we scroll down, we can see here that this download included records from 43 different data sets. So any of these institutions are able to actually see that their data have been used in this particular publication. So let's just pull as an example. This is UC Santa Barbara Invertebrate Zoology Collection. So they have 34,000 records in GBIF, and we have found 179 citations. So these are cases where data was downloaded and used in papers where at least one record from this particular data set was included. They can then go ahead and click through to that and see, oh, here on the top was that paper that, that I showed before. So let me go back to my presentation. Here we go. All right, so just a bit of background and history on how this has been going. So we started in 2015 assigning DOIs to data sets and downloads. This has been a slow progress. Uptake has been very slow, but now we are at a point where DOI citing papers basically make up all growth and citations. This has not been easy. This has involved a lot of chasing directly corresponding authors, editors, journals, and so on. But I'm very happy to say that so far in 2023, papers with DOIs make up nearly 60% of the citations that we encounter. So that's it for me. Thank you.
Okay, I think I get to go next. So my name is, confirm everyone can see slides and hear me okay before I jump in.
Yep. Yes.
Cool. So my name is Alicia Charleston, and thanks to the organizers for the opportunity to come and speak to you today. I'm kind of on the tail end of some of the other talks for a good reason, because I'm going to talk about in our system why the data needs samples, and then of course persistent identifiers. So quick overview of what Kbase is. I'll try to run through these fairly quickly so that we can talk about the more interesting stuff. Kbase is a community-driven research platform for systems biology. So we really work to integrate both tools and data to accelerate analysis, support collaboration, and then help publish discoveries. We sit under the Department of Energy, Biological and Environmental Research Office in the Office of Science. And on the left is kind of a generic diagram of what a data management lifecycle might look like. So people collect samples, they send those samples into some of the BER resources, such as the Joint Genome Institute, which Joan mentioned. Those samples get turned from physical sample into data, and then that data could get put into Kbase, for example, where people would analyze it, do some visualizations, make some inferences and conclusions, and then work with places like ESS Dive, which Joan is from, to curate the data and get it ready for publication. Kbase kind of sits in the middle of that. So we take data from several different places. We allow users to integrate that data across the different sources, give access to a lot of tools to analyze that data, build their own analyses, and then share those reproducible analyses with collaborators, and then start to publish their results. And I'll get into kind of like why the sample information is super important here in a second. But I wanted to just give you a little bit of an overview of kind of what the interface looks like. So Kbase is built on top of kind of a Jupyter notebook structure, but we provide a graphical user interface so people don't actually have to be able to run command line codes in order to be able to use our platform. We focus mostly on biological sciences, especially omics data. This is kind of a GIF that sort of shows what this reproducible structure looks like on top of the Jupyter notebook. Anybody can access it. All you need is access to a web browser. We provide all of the compute, and on the on the left-hand side on the top lists all the data that are in this particular narrative, and then it's a list of all the tools that we make available to the community. And these tools are basically wrapped into Dockerized images that people can use to run. We also make it easily for people to then take their reproducible notebook and share it with either individuals or share it with a broader organization that they might be part of, their research lab, for example. And then they can make it public and share it with our currently over 31,000 users globally. So when it gets to publishing results, we're calling these FAIR narratives. You can take that reproducible notebook and get an HTML snapshot that is now indexed and available outside by search engines. There's an example link here at the top that kind of gives you that resolvable link that it goes to. We then can actually get a DOI. We leverage OSTI, the Office of Science and Technical Information, which gives DOIs for all of the Department of Energy projects. We give them a DOI that sort of maps directly to the same URL for that HTML snapshot of their narrative. This makes it sort of fair, so it's findable, accessible, interoperable, and reusable a bit easier. And we also track full versioning of that. So on the screen now is an example of a FAIR narrative. So the narrative itself is called the geochemical and sequencing data from this particular paper. The paper is linked below that, where the person actually took the DOI from the FAIR narrative and then cited it in the reference section of the actual publication. So now we're able to link the DOIs to each other, the publication DOI and the dataset DOI. And then on the right, I've provided an example sample table from that particular narrative. And it's kind of hard to see, it's pretty small, but there's an identifier, latitude and longitude, depth, and various other metadata associated with that. Things that we've talked about, just go into sort of describe the physical environment that the sample came from. Digging into that a little bit more, this is kind of what it looks like when you go into the actual narrative. Again, there's the title of the narrative at the top. And then when you look at the actual sample set from that narrative, you can actually look at that sample set on a map. You can get a summary of the samples that were in there. These ones were sediment. There were 48 samples. They have 7 attributes that are associated with it. And then it talks about all the actual data that is now linked to those samples. So all that sample metadata can then be run in analyses. For example, this is looking at an amplicon matrix. So this is basically looking at taxonomic distribution. In the taxonomic distribution down here at the bottom, gives counts and mean of the taxonomy in each of those samples with the sample labels across the top that came from that nice curated sample metadata template. And so Kbase is really working with our users to understand how to combine things like sample identifiers, all of the rich metadata that comes in with those samples to describe the environment, such as temperature or materials that was collected from, the depth, anything people put into their experimental design, and then combine it with the actual data that comes from those physical samples. So maybe it's chemistry data, maybe it's omics data like genetic sequencing, or maybe it's taxonomic data that they've done, and then provide them enough tools and visualization so that they can actually recombine those samples in a way that makes sense, all in a reproducible format, link it all together, and then start to do some interesting correlations about how the samples are behaving based on the data that they've actually contributed. I don't need to preach this to this audience, but one of the things that's really part of this conversation is just making sure that the research effort that goes into a publication becomes more visible. So people put a lot of work into proposals and protocols, and then the samples, and then taking the samples and turning it into data, and then processing the data, doing the analyses. And so a lot of the effort that we're trying to do in general across all of the PIDs work is to actually make sure that that research is more visible, and it's not just the publication that gets the credit for it. So the other part that Kbase is really working on just in general is persistent identifiers across all of the data products and samples that come into that, Once those all get imported, so a researcher imports their data, they bring in sample identifiers, they bring in public data, we can actually track provenance of all of those things in system and ensure that credit gets produced. So here's an example. Orange researcher brings everything in, creates a nice fair narrative, gets a DOI for it. And then the blue researcher finds that and says, hey, that's really cool. I want to compare that to my data. So they make a copy of that. They import their own data. And now we have this knowledge path that we've actually can follow. And we actually automatically cite all of that. So any of the samples or identifiers in the orange narrative, when the blue researcher makes their fair narrative, we auto cite all of those persistent identifiers in that second DOI. So it starts to really build out the knowledge graph of how all these research products are associated to each other very quickly. All of the DOIs that OSTI gives us are automatically contributed to DataCite. If somebody cites that DOI in the publication, it makes an auto link to Crossref, and then we're actually putting in some of the background infrastructure so we can start pushing these records to people's ORCIDs. And the idea for that is that now they have a visible record for when they go for promotion of tenure or applying for jobs of everything they contributed, not just the publication citations. And this is an example of kind of the convoluted path that researchers often have to take to do this. I actually stole this linkage from a slide that Joan had put together, where there's a sample core that has CSAR identifiers associated to it. That sample core then goes through some processing to turn that physical sample into data. The data goes to two different BER resources, so different types of data come out of both of those, and then those have to then get combined into Kbase along with the actual sample information in order to give us enough context to then hand it back to Joan, say, hey, this is then ready for publication. So it's a lot of still working with researchers throughout this whole process. We're looking how to automate as much of these transfer of this information as possible. But there is quite a bit of work still done to actually streamline this as much as we can. So with that, I'll go ahead and just put up a quick by the numbers from our last quarterly report. Kbase has been used in about 500 publications now. We've got over 31,000 global users, and we currently have over 600 terabytes of user data. And that's the team. So I'll stop there.
All right. So how much time do you have left?
25 minutes.
We have 25-ish minutes. And in those last at least 20 minutes, we wanted to open up the floor for questions. So I think let's start with any specific questions for our speakers. I saw some in the notes, but I wanted to give folks a chance to just ask them out loud. Yes. Oh, can you?
Well, I guess somewhat question, maybe a few comments. I mean, I'm really excited to see this because I've kind of left the samples world for a while. I was involved in developing the IGSN and all of that years and years ago, but kind of have not touched it too much. I wasn't really able to provide a lot of comments in the quick format that we had to go through that because I'm not familiar with the guidelines document. But I guess a couple observations about that. It occurs to me that it might be useful to maybe split that concept up into sort of three parts. I mean, within the theoretical guidance, you have a mix of kind of the theoretical basis for this and why we're doing that and the argument for it, and stuff that's more like a checklist and a protocol and like this is what you actually follow and do in some way. And the protocols are really something that I think is going to develop iteratively over time and need to be versioned over time and come from different parts of the community that are implementing the ideas and carrying that forward. And I think it would be useful to maybe really get specific and clarify some things in that guidance document to lay out why this is really important and what needs to be done. And one of the big pieces that's missing in there, and I guess this is a little bit to something that struck me with Dave Viglas's comment about, well, the most important thing is we just need to connect the paper to the sample. I would argue that it's a bit more complicated than that. We need to know what's going on there. I mean, what are you actually citing when you're connecting a paper to a sample? We need to understand the context a little bit more there. And, from the GBIF talk as well, that's going to be somewhat complicated. There are times when you're going to be looking at, hundreds of samples in a given paper, and you need to be able to dynamically connect to a list of all of those, a data set of all of those samples, maybe from across multiple repositories even. Each of them is going to have their own identifiers and everything with that, but you need to be able to point to that dynamic collection of a bunch of samples and provide clarity in the paper about what it is that you're actually talking about there. Was it a set of observations and measurements taken from those samples that you've brought into your analysis that you're using in some way? So the paper to samples connection, I think, is going to be more complex than other things, and it's different than anything else. I mean, when you're talking about samples in a paper, it's different than citing another paper. It's different than citing a data set. It's a different kind of dynamic, and I think that needs to be clarified in there. So I think that really can make up maybe more of the bulk of the paper, and then if you maybe provide then that framework where lots of different folks who are implementing the ideas in that paper can contribute specific checklist protocols that, the specific ways that we're going about it for a given circumstance, for a given set of collections, maybe based on what's laid out in that paper. I think that would be, a really helpful way to do that. And then a third piece might be more of a flashcard, one-page thing of, right in your face. This is why you really need to pay attention to this. These are the citation guidelines that should be followed as you're going about the process in some way. So anyway, a few comments, but overall, I think it's a cool thing. I'm excited to see where it's going, and thanks for the work on it.
Thank you. I do hope that you are interested in helping us write some of those very helpful comments down. And we will bother you to contribute. I do think in the full, so we try to present a small kind of like high level summary of what the guidance is so far. And in the full paper, we do try to split that out, but that is helpful to think, to know that maybe some further splitting of that out would be useful. Anne.
Yeah, I would like to hear more about these reliquary documents and what kind of, can I get some examples of what might be a reliquary document?
Yeah, Dave, do you want to answer that?
Sure. Well, first up, I didn't come up with the concept of the reliquary. Maybe Andrea can provide a bit more background on that. But
and Leslie is here also.
Leslie.
We can put her on the spot too.
Yes, Leslie's can answer that. But from my perspective, the reliquary is it's really an opportunity. It can, at a minimum, it contains a set of identifiers, and by bundling the set of identifiers into a document, there's all sorts of interesting additional information you can provide with that. So with respect to the previous question about the context of application within a publication, this is an ideal location where you can provide a whole bunch of context about why specific identifiers were used and what purpose they're used for and so forth. Without embedding all that information directly in the publication, you can add it to the reliquary document. So from a technical standpoint, from my perspective, a reliquary document would basically be what's called a named graph in the RDF world. And it would essentially, at a minimum, contain some basic metadata about the purpose of the document and the set of identifiers to the material being referenced.
I can put a link in the notes. There's a few of us, particularly Shelley's been leading this for some time with the AGU Complex Citations Working Group. And there's now a new RDA working group set up on complex citations. So you can join that. I'll try and put the contact. But the idea is that most journals will not accept more than, say, 20 objects or DIs in a paper. So the idea is that you set up a reliquary, which is a terrible name. If we can get a better name, we will, but we can't. But a reliquary is a bag of bones. And it itself has a DOI, but then within that you can have hundreds, maybe even thousands of individual objects, each with their own DOI. And so you know what the component parts are, say in a model, like a climate model. Martina Stockhouse is leading this a lot. So I've developed a model, but what are all the component input data artifacts? The samples, it's the same thing. I can go on a trip and collect, 100 rocks, 20 blue, 20 black, 20 green, and I can do a paper on the pink rocks or the green rocks or the pink and the green rocks, but it enables you to preserve those combinations. And what's particularly important, particularly for samples, is that often the researchers who collected the individual samples, so when they get put up into a collection and the collection just gets a DOI, they lose all credit, all tracking, and it's really hard for museums to show the value of what they're doing. So yeah, I'll certainly, Shelley, if you put the link in the notes yet, or one of us will get the link to the RDA working group, and please join us, because I've actually put a placeholder there for samples. And if someone wants to help work that use case up, I'd be really grateful, because I've got 2 others and I can't do all 3. So does that make it clear to people what a reliquary is and why we're bringing them in? And Shelley, do you want to add anything? Yeah, it's so that you can draw a graph and trace and really know who put what in. And more importantly too, if you've got full metadata and you've got the funders, then funders can actually start to track the impact of their funding of, say, a particular sampling expedition, or, as I said, a museum or repository that's struggling for funding, but they'll be able to track their samples more carefully into every place that sample's cited.
Might be a little bit excited that Dave had the word reliquary in his slides and I was taking pictures so I could tell the team that we've made it to ESIP. So this is really exciting. And based on the description, do you see a use case there for your work for a reliquary?
For a reliquary?
Yeah. Having to do with samples.
My current work doesn't use a lot of samples, but yes, I definitely agree that
Okay.
having a reliquary is like a named graph that links everything together so that you can collect metrics on, who did how much of what, which samples contributed what to what paper. Yeah.
And one of the key elements, and just to add 30 seconds, just to add a piece to what Leslie was saying, the key element here is that many of you know, like DOI collections. So think kind of like that, but credit didn't work because the children of a DOI aren't getting automated attribution and credit. So this is fixing it. Yeah. So we picked a word that no one else was using, so you wouldn't assume, except maybe the Christian church, and in the sense that we would try to fix that, and then I don't know what we'll call it, but reliquary just made it to ESIP, so there we are.
I think reliquary is probably sticking. We all think it's a little goth. I like it. Yeah. I have a question if nobody else does for Daniel, and I'm gonna just ask it. So, and I saw somebody else put this in the notes, and that's why I'm asking it. And especially given what Shelley and Leslie just said, is there a reason why we can't backtrack to specimen citations from the data set citations in GBIF? And is that an interest that GBIF has?
That's a very good question. The basic or the simple answer is that we don't have at the moment entirely persistent identifiers for the individual records. So that's the simple answer. But this is definitely something that we are interested in and that we are working towards solving in one way or the other.
Okay. Go ahead.
Yeah, this is Matt Jones from UC Santa Barbara and NCs. I'm curious about this reliquary concept and how it relates to other aggregation standards that are out there. So in particular, what are the similarities and differences to OAI object resource embedding resource maps and to the data package.json standard for aggregating parts of a data package? So both of those seem really similar to me.
I see Leslie volunteering to take that on.
So I've put the link to the working group in the notes, so please join, Dave. We'd love to hear that. The other one is that, like, I hadn't heard of those two, so we'd love to hear about them. Shelley, had you heard of those two? Yeah, okay, right. I must have fallen asleep. The meetings these guys have from North America are at terrible times for me. But the other thing too is that also, as Shelley said, with DataCite DOI, you've got a collection and you can kind of do it through that. But to our knowledge, correct me if I'm wrong, that's the only PID system that you can do this idea of a collection of, you know, multiple DOIs together, multiple citations. But yeah, we're trying to work out who else is doing it. DataCite have joined our working group, and so yeah, I mean, it's just there's a very big need for it, and we're trying to see if we can utilize an existing system that can be accessed by all, which is kind of a part of the issue with the using the DataCite DOI collections.
So I'll just add in that I think the OAI ORE spec is pretty much exactly a reliquary. It's you know, basically a RDF definition for related resources, and I think it'd be super useful to leverage all the work that's already gone into that sort of infrastructure. And similarly for the JSON package, I'm not as familiar with JSON package, but I think there's also a lot of parallels there.
So who creates the reliquary? Would it be the person who is archiving the samples or the person who is using the samples to publish a paper and then needs to cite them in their paper?
Go ahead, right behind you.
In my opinion, it would be the person who is pulling resources from multiple sites together to go with a publication, because whoever is archiving the sample would hopefully be giving the sample its own individual DOI. And it's when you put samples together, and that's partly the other driving need for the concept of a reliquary, is that sometimes people can describe sample collections that actually come from multiple repositories or museums. And so that's the important thing of getting down to the granularity of each sample and child of that sample having a DOI, each one. And then it's your aggregation. So the same sample can be in multiple reliquaries. It gives you that flexibility to create multiple combinations. Another use case we have is, for those of you, sorry, geologist, geochemical analyses. It's the same thing, you do a lot of individual analyses that you can put together in multiple combinations in multiple papers.
So I know this is the physical samples group, but thinking about it from like a publishing standpoint, so we're doing this with like physical and a lot of, are you guys working with any groups that are doing like web services and those kinds of data, or is anybody else playing in both spaces?
What do you mean web services?
We're having this like with digitized maps.
Oh, I see.
And the same kind of like you've got so many layers of
No, we're not in short, not really.
Okay.
We've been trying to sort through, like, working with the physical digital stuff is hard. I truly think that trying to figure out physical sample citation is harder than just data citation. That is the conclusion we have come to over the last year, that this is just really hard because there's always going to be this fundamental disconnect between the thing and the data. I do think that collaboration with those groups would be important though. And if you would like, I'm just going to keep telling people to come to the cluster.
Okay, yeah.
But like, yeah, please share any groups that we should be aware of, and please add your email to the notes.
Okay, thanks.
Yeah, in the back.
This is in a way similar to the NIH's NCBI database in that you create a project, and then inside that project you can add samples, and then each sample can have a data set or multiple data sets hooked to it. And if you want to create a new project, you can still pull those old samples in with a new objective. Not that NCBI's way of citing their samples is a good way, but this idea of a reliquary has been done before, just with different names.
Yeah.
And it seems like if it's been done before, it should be able to be done again, maybe better with some improvements.
I agree. Yeah, that's helpful to, that would be a helpful model to look to, it sounds like.
I might have missed this in the run through of the sample sharing guidelines. But it like it's part of your point that you were making about the difference between data and samples. Made me wonder whether you thought about, you know, sample availability statements as like the way to do this versus tables or supplementary figures. It seems like there's a lot of options that you can take through these guidelines. And like, it seems like maybe having just the best practice or what the leading practice is might be really helpful to kind of define and what that might look like.
This right now, well, I mean, Joan also, please, and Val, who's online, and anybody else, but like this is the hardest thing to figure out right now. I think this is like, we feel like we have a pretty good idea of the general metadata that should be included and the general practices, but where to put that information right now has kind of been the hardest part. And so I was talking to Shelley, we're going to be reaching out to AGU. A lot of it comes down to the publishers, because I think that if the publishers say, here is a place to put this information, and we will not give you page limits, and we will surface the metadata, that makes a bunch of our problems go away. And that might just work for AGU publications, but our hope is that that could be a leading practice that other journals would follow on. And I do think it has to come from the publishers, because I know that collections have been asking people to cite their samples for decades, and they can't even get an acknowledgement statement. So it has to be something that's like required before you can get your paper out there. So yeah, that, and go ahead, Joan, or.
I'll just add that that was a great response, but I think it will vary a bit based on the community. So like the natural history collections, it might be more suitable to do something like a sample availability statement in a paper. But I think for other communities that are dealing with like very large numbers of samples and working with different data systems, it's more important to actually have a sample data set. So that's why we have a couple of different options for different use cases.
And I think we've got like 4 minutes left. So I'm going to go up to our next steps. So to like, to that end, we need use cases and examples. We need volunteers to take their data, to bring their data and go through the guidance with us and try to make a sample metadata file and really practically think through how would you adhere to these guidelines, what does or doesn't work, because there is such a wide breadth of different kinds of samples out there. I'm very limited in my context of natural history collections. So we need more people to bring them. So for the millionth time, please give us your information and contribute. And we're also going to be working on a paper on the needs and current efforts towards sample use tracking. So not just the guidelines, but a paper summarizing what we need to do next, because the guidelines are only one part of the puzzle. Joan, do you want to say more about this? Okay. I think so. Yeah.
It's not sharing.
Oh, it's not sharing. Okay. That's right. Sorry, guys. Yeah, I think last question for everyone. Are there any other next steps we should prioritize as a group beyond getting these guidance done, writing this paper, and working up test cases? Is there anything that's standing out that our cluster should try to take on?
This was huge.
Thank you. Abby and then Anne.
Okay, yeah, I just am wondering about the social engineering that you know, because as Daniel mentioned, it's taken a lot of his time and effort to get people to even cite one DOI in a paper. So to create a reliquary, you know, it's just not like, how do we socially engineer that? It sounds like maybe you think making it mandatory to get published is the way to do that. So maybe you already have something planned, but I'm wondering about that.
That's a good thing for us. I think that's a great suggestion for us to consider. I think that's one piece of the social engineering, and I think maybe some actual engineering of tools to make that possible could be helpful too.
So following on from that, I think that making the reliquary, like you said something about what tools should be the priority, like something that makes it very simple to generate one of those. And along those same lines, I also think that not just talking about this like minimum metadata standards for physical samples, also looking at the very specific structure of the named graph and just get that out there as we're using the provenance ontology, or we're using this type of identifier, we're using this vocabulary, whatever, just have that done up in the very beginning so you save yourself a lot of pain later.
Okay. That's helpful. All right. Oh, did you have a question? Yeah. Comment.
So I run a few data repositories on sample-based, and we have, you know, our data sets include most of this sample information already. So would that be sufficient? Like if we have, you know, the other measurements that went with the measurements on the samples have the metadata for the location and their, you know, IGSN and so on and so forth. I mean, is that sufficient? Is that sort of?
That's a good question. So so basically create guidelines for repositories, it sounds like, for.
Yeah, I mean, we have like, you know, rock dating or
Yeah.
paleomagnetic, and when there's a sample, then we collect all that information already. So I don't know if we need another list.
My guess is that that probably is sufficient as long as people can access that information, which is the tricky part sometimes.
Okay.
Yeah. All right, we're right at time. We are supposed to come up with 3 takeaways of this session from this session. If you have one, please tell me and I'll write it in the notes. Thank you all for being here. And for the millionth time, please join our cluster, share your email. Anybody want to do a Funding Friday thing? Come talk to me. Yeah.